Hello, my name is Stepan Nersisyan. I'm from Higher School of Economics University, from Faculty of Biology and Biotechnology. And today I will speak about a pipeline for feature selection and classifier construction and its application to the problem of breast cancer recurrence prediction. So classification is widely used in several biomedical applications. For example, some people use it for construction of prognostic gene panels for cancer. For example, we can build some gene signatures which can predict recurrence of some kinds of cancer. So the biggest problem for biomedical classification uh, is that the number of samples is usually low. Number of samples derived from tissues, from tumors of patients. Usually it's tens or hundreds. And also we usually get very high dimensionality. For example, if we analyze some gene expression data, we usually have about 10,000 or 20,000 genes. So this situation can simply lead to overfitting. And today I will speak about our approach for preventing it. So we focus on predicting of recurrence of ER-positive breast cancer using gene expression profile in primary tumor. So the following scheme was developed for classifier construction. At the first step, we perform unsupervised feature preselection, thus reducing the number of genes analyzed in the next steps. Then we use supervised feature selection to select some lower amount of genes. Then we perform exhaustive search of all fixed length feature combinations from genes selected at the previous step. Finally, for each combination, we feed support vector machine model and test its performance on independent validation set. So we use several data sets to test our pipeline. They included several microarray data sets from different Affymetrix platforms and RNA sequencing. So here's a table. We use 5 data sets from Affymetrix U133A, 2 data sets from the next generation of Affymetrix arrays, and RNA sequencing data from TCGA BRCA. So here you can see geo-accession IDs and number of patients in each of classes. So first class is composed of patients with recurrence during the first 5 years after surgery, and the second one is composed of patients without recurrence with at least 7 years follow-up. And first, I would like to speak about unsupervised preselection. So the idea is that genes with high activity at intracellular level can have big prognostic potential. So for that, we perform miRNA gene TF regulatory network construction, um, composed of 2 steps. And the first step, we build the network using database level information. And the second step, we incorporate information about miRNA or mRNA expression in a set of patient tumors. Thus, we can achieve simultaneous sensitivity and tissue specificity. And the source codes of our tool are publicly available on GitHub. So these are some pictures of the network, on the breast cancer interaction network. Is a global overview, and there are some fragments of the network. This is a strongly connected component, and here's a component composed of highly reliable prognostic genes. From the point of feature selection, we used 3 methods, including selecting most differential expressed genes, L1 regularized models such as logistic regression, and then we selected random gene subsets. So here's a brief overview of our pipeline. So we have all genes or genes from the interaction network This is a pre-selection step. Then we use feature selection. In this image, we take n most differential expressed genes. Then we iterate over all k element combinations, feed SVM classifiers on 2 microarray datasets, filter them on other microarray datasets, and evaluating performance on validation set. So our results include the following points. First, pre-selection is very important because it allows constructing of cross-platform classifiers. Then we observed that selecting most differential expressed genes is the best technique for feature selection. L1 regularized models were highly overfitted, and random gene selections led to higher rate of underfitting. So, for example, here's an 8-gene signature we built using the pipeline. Here are genes. Some of the genes were already mentioned by other research groups, for example, FOXM1, RECGAP1, and others. Some are not, for example, ERIC2, and others are not available in existing literature as an examples of prognostic markers for breast cancer. And quality characteristics are very, very high. For example, area under the ROC curve was equal to 0.78, and sensitivity with specificity were higher than 0.67. So here's a ROC curve, and here you can see a Kaplan-Meier curve with p-value with statistically significant p-value. So as a conclusion, we saw that interactome-based feature preselection is a very efficient technique for feature preselection for reducing amount of computations initially. And we see that different feature selection algorithms show completely different performance in terms of overfitting. And one more time, once again, we demonstrate that exhaustive search over all possible gene combinations is a very efficient technique for prognostic combination construction. Here are references to our previous works on that topic. So thank you very much for attention.